The Beckman-Argyros Award in Vision Research was created in 2013 by the Arnold and Mabel Beckman Foundation in honor of the friendship between Arnold Beckman and George L. Argyros. The award recognizes individuals who have made transformative contributions to vision research and provides recipients with support for future research. Dr. King-Wai Yau of the Johns Hopkins University School of Medicine has been selected as the 2019 recipient of the Beckman-Argyros Award in Vision Research for his groundbreaking discoveries related to image and non-image vision.
My entry into vision was really accidental. After my graduation with a Ph.D. and then starting to look for a postdoc position, my Ph.D. mentor, John Nichols, emphasized the greater importance of finding a mentor who has a very rigorous approach to science. So taking his advice to heart, I went to work with Dennis Baylor, and he happened to be working on the retina at the time. And afterwards, Baylor said, look, my former mentor, Alan Hodgkin, is truly an extraordinary scientist. You'd be good if you go work with him. I worked side by side with him for about 1 to 2 years, and that's how I got into vision.
Well, I think King is an outstanding recipient for this award. It's a perfect choice. If we go back and think about what Arnold Beckman stood for.
Described by George L. Argyros as brilliant, kind, and generous, Arnold Orville Beckman was truly a pioneer in his field. During his tenure at the California Institute of Technology, his research transformed into a business opportunity in the form of chemical instrumentation. He permanently changed the world of scientific research through his revolutionary inventions, many of which paved the way for common scientific instruments used today, such as the pH meter and the spectrophotometer. As lifelong philanthropists, Arnold and Mabel Beckman donated millions of dollars to scientific research. In 1977, Dr. and Mrs. Beckman founded the Arnold and Mabel Beckman Foundation to support chemical and life science research.
I think the most important, to me, single aspect of King's character is really his devotion to answering basic questions about sensory biology.
The question of how light energy is transduced into an electrical signal has been in debate for about 15 years. And when I set up my lab around 1981, I began to look into this problem, to some degree also in conjunction with Yevgeny Fesenko's group in the former Soviet Union. We were able to sort out the 15 years of debate into a coherent picture, and it involves 2 second messengers, that is small molecules in the cell that mediate signaling. And we established that cyclic GMP mediates transduction, that is conversion of the light signal into an electrical signal that the brain can understand, and calcium ions that turns out to be very important for mediating adaptation of the photoreceptor.
His contributions have focused largely on the physiologic properties of photoreceptor cells, but most importantly, he's brought a level of rigor and precision to that area that has been unique.
There are a lot of diseases. Practically every protein involved in this process of phototransduction in rods and cones can create disease when mutated. And so knowing how each protein's role is in the process allows people to start thinking about how to make remedies.
One of the very unique and special features of King as a scientist is that he doesn't listen to other drummers. He goes by his own drummer. So he follows science, he doesn't follow trends. He really knows what's, interesting, very basic questions.
So for over 100 years, it's been thought that rods and cones are the only light-sensing cells in the retina. But now there appears to be yet other cells that also serve to receive light.
King is a unique member of the Johns Hopkins family. He's an outstanding researcher, he's an outstanding mentor, And he's done his own thing scientifically. He's very much his own man in terms of deciding what areas he thinks are important, and he's pursued those relentlessly, and I think that has set an example for everyone else in the community.
So I'm continuing on with my studies on the phototransduction mechanism in rods and cones, and also the phototransduction mechanisms in intrinsically photosensitive retinal ganglion cells. Additionally, we are venturing into a new question regarding the light-dependent orientation of the rod and cone photoreceptors in the retina. So I'm very grateful to the Arnold and Mabel Beckman Foundation for this award. It will allow us to hopefully get into some further exciting work.